Hello, good morning, good evening, good afternoon, depending on where you are. Welcome for this to this insight session on developing COVID-19 vaccines. Unfortunately, COVID-19 needs little introduction to anyone. The pandemic has brought the world to its knees for the best part of 2020. And while we have seen that lockdowns and distancing work, and treatments such as dexamethasone have helped us manage the disease to some extent, overall infection rates and the death toll continue to be intolerably high. And worryingly, we continue to learn about new sequelae of the disease, some of which could be very long lasting. Right now, all eyes are on vaccine development. Out of 300 candidates in the works, around 40 are currently being tested in humans and 9 are in phase 3 clinical trials. The pace of development has been unprecedented. One of these 9 vaccines is being developed by AstraZeneca and the University of Oxford, another one by Johnson & Johnson. I am Magdalena Skipper, Editor-in-Chief of Nature, a weekly journal of science, and I am fortunate to have with me here for this COVID-19 vaccine development session, Pascal Soriot, Executive Director and CEO of AstraZeneca, and Paul Stoffels, Vice Chairman of the Executive Committee and Chief Scientific Officer of Johnson & Johnson. Welcome to you both.
Thank you.
So let's start. Welcome. Let's start with the basics. Both of your approaches to COVID-19 vaccine development rely on adenoviral delivery. Can you give us a brief overview of each of your vaccine approaches and how similar or different are they? Let's start with you, Pascal.
Well, I mean, they are very, in many ways, they are similar. As you know, there are several technologies in development. One is modified RNA, and the other one is this adenovirus vector, which both Paul and Paul's company and our company are using. Ours is based on a chimpanzee adenovirus, and the J&J one is different. So, Paul, maybe you want to talk about your own vaccine.
Well, it's based on adeno26. It's a human adenovirus, and it is using the cold virus, which is not replicative anymore, to carry a piece of the COVID virus into the body, the genetic information. And the genetic information is then coding for producing the spike protein in the body of the human. And that is a mechanism that we use the adenovirus as a carrier to bring a part of a genetic part into the body, and that is producing then the protein to get to the vaccine. It has proven both for AstraZeneca as for us to be a very efficient way of doing that, and that's what we use now to develop very quickly vaccines.
So as you can see, Magdalena, the technology is very similar. The difference is we both use an adenovirus vector that carries the spike protein, but the vector is slightly different.
Yes, thank you. And then, of course, both of your companies now have gone into phase 3 trials. Which populations across the globe are these trials running in, and what determines your choice? I ask because, of course, we are acutely aware that different populations differ in the susceptibility to COVID-19 and then indeed in their disease manifestations. Paul, perhaps you can tell us first.
Yeah, in the first phase, in phase 1, we study in healthy volunteers to know the safety and the risk, but also the immunogenicity in a let's say a clean situation. But in phase 3 now, we go very broad into and mainly targeting the populations which are most vulnerable. That means the elderly in the US, the African American, the diverse population, but also we're studying in Latin America and in Africa. So at the end of the study, we should have a very broad and diverse population into the study, which are typically the target and those people who are most vulnerable to the disease.
And Pascal, what about AstraZeneca's?
Very similar. I mean, our development program is structured a little bit differently, but fundamentally we do the same. We have very diverse populations in Latin America, in North America, in Africa, and also we have a study in India. So we try to go after very diverse ethnic populations, but also we need to look at different age groups. We go 18 and over. We haven't got any pediatric study yet, but essentially we try to cover different populations, ethnically, but also age-wise.
Mm-hmm. And so I understand different trials are aiming for different population sizes in which the trial is ongoing. How do you determine the size of your trial?
Well, the size of the trial is mainly determined also by the transmission, the rate, because you need to go to high risk areas where there is a lot of transmission in order to be able to show protection. And we measure two things, is protection against disease, that's the primary endpoint, and protection against transmission. And in order to have a chance to reach a significant, statistical significant endpoint and have enough data to submit for emergency use and later on for BLA, for full approval, you need to have quite significant numbers. And we have done a lot of data science, epidemiology to see on where and how we have to do the study to target the people most at risk. And we determined for a phase 3 study that will go up to 60,000 people on a global basis to be able to show the safety and the efficacy.
Essentially, we follow, we all follow the same approach. You have to define the size of your population based on the infection rate in the communities where you're going to run your trial, and also how fast you would like to get a response, and that define the size of your study. But you also need to have a sufficient safety database, so you need to vaccinate a sufficient number of volunteers to have enough safety. Typically, it's around 50 to 60,000 people that need to be vaccinated, so you feel comfortable you've accumulated enough data.
Mm-hmm. And so, am I right in saying that the efficacy of the vaccine you and others are aiming for is around 50%? And experts say that this may not be sufficient to achieve herd immunity. And how does this compare with the FDA requirement and other vaccines against other diseases? And what does it mean for transmission and indeed the need to revaccinate in the future?
Well, the minimum level has been set by the authorities at 50%. But of course, we aim at the most effective vaccine. We optimize for potency. You can use boosting. So there are different mechanisms for a much higher. So the expectation we have is that it is significantly higher, but we don't know. At the moment we are testing, and that's where the minimum level is at. But we should aim for higher protection, and hopefully we get to that.
There are different numbers that are floating out there, and of course that confuses people a little bit. First of all, as Paul said, you have the minimum threshold established by the regulator. So for instance, the FDA has said they need a minimum of 50% efficacy, and the lower bound of the confidence interval statistically has to be 30% and above. So that's what the regulator defines. Then you have the rate of efficacy that you use as an assumption in your statistical analysis to define the size of the study and when you can do an interim analysis, etc. And then there is what you hope for. And we all hope for more than 60, 70%, of course, but the minimum is 50. And depending on the protocol, different companies use different numbers, but I think mostly people have used 60% efficacy as an assumption in their protocols. Well, we all hope for more than that, of course. Yeah.
And so can you comment on the second part of my question about transmission and the need to revaccinate? How are you projecting that? Will we be looking at repeated vaccinations?
We have to learn that, but most probably yes, and that's also depending on how the immunity evolves in humans. Yeah, we don't know if you generate antibodies and cell immunity, how long that will protect you. We know from animal models. We have studied that very extensively with other diseases that we can get longer term protection with a single dose, but ultimately you need to boost for longer term protection. So we'll study that in several different parts of our study. We'll do a boost at 2 months, and we'll then learn whether we do a boost at 6 or 12 months, but boosting will need to happen at certain points for long-term protection.
The big question that, I mean, first of all, of course, we don't know whether those vaccines work, but we all hope they do. But then the next big question will be in how many people and what's the percentage of people are protected. And then the next one will be how long are you protected for? And that answer we will get over time.
Yes, exactly. But Paul, do I understand correctly that the Johnson & Johnson vaccine is designed to be a single-shot vaccine? Is this correct?
Yeah, that's correct. We will test both a single shot and a booster shot, but we developed the same vector in 4 different indications. We are working we did a certain development up to a certain point in Zika where we used a single shot. Also, RSV now is in phase 3 with a single shot, but Ebola is a boosted shot because Ebola is a very lethal disease. We used a prime boost, even in heterologous prime boost with 2 vaccines to make sure that you had one as close as 100% protection because of the lethality of the virus. And so in Ebola now we are vaccinating over up to 200,000 people in the Kivu and the Rwanda area to protect for Ebola. And that experience told us that if we developed in a certain way with certain insights and optimize that, COVID inserts, we can get to a single shot. We showed that in an animal model, and now in phase 1. And hopefully now we are going to study that, not hopefully, yesterday we announced we are going to study this in a phase 3 study. Later on in the year, in October, we'll start a boosted study once the phase 1 data will become available.
Right. And so if I can just follow up on this, so is it fair to say that whether you are able to design a single shot vaccine or a booster, go for the booster design, is determined by a combination of the virulence of the virus, but also your ability to target, create immunogenicity?
Well, it's also determined by the vector as well as the insert. So there are many factors which determine the potency and the immunogenicity, but that is work from the scientists over many years, which in our case was the collaboration between Dan Baruch at Harvard, our team and in collaboration with NIH and the vaccine team in Leiden. So they worked on already 10 years on this topic. Hopefully with success. We'll see soon.
Sure. So on September 6th, AstraZeneca's phase 3 trial was paused after a person who participated in the UK trial experienced an adverse reaction. The trial was, of course, restarted on the September 13th. And as we reported the day later in Nature, the scientific community was not overly concerned about the pause itself, which in fact in large trials is not uncommon, but there were criticisms associated with a lack of transparency about the reasons to suspend the trial and the process that led to it being resumed. Medical confidentiality was cited as a key reason, but some have of course argued that information could be disclosed in a way that avoids individual identification. So Pascal, could you shed some light on the decision-making here? Isn't transparency important for public trust? We need the general public to want to receive a vaccine when it eventually arrives, right?
Yeah, no, absolutely. I mean, first of all, I should say that stopping a trial in a vaccine program is not uncommon. I mean, it's not very common either, but it happens. And if you place safety at the center of everything you do, of course, you're going to have to stop and look at events, and that's what we did. And both the Independent Safety Committee and the regulator allowed us to restart the study. Now the question is, how much transparency can you provide, right? And typically, you don't provide information. I mean, the guidelines, I should say, the clinical guidelines recommend that you don't disclose patient-level information, and also you don't disclose much information at all because you could compromise the study. So that's basically what is the background from the guidelines viewpoint. Now we are actually looking at how much transparency can we provide, considering we are in a very special set of circumstances, of course. So we've disclosed our protocol. And we're discussing with other companies as an industry, what kind of transparency could we offer without compromising patient privacy, of course, but also without compromising the trial itself. Because if you disclose the information, too much information, you can actually compromise the clinical trial itself.
So did I, excuse me, did I hear correctly? You were saying that you're discussing it with other companies. So with a view to arriving at some kind of a consensus?
Yes, we're trying to kind of come up with a consensus to say, okay, well, you know, this is what we feel is reasonable to disclose. That is typically not disclosed, but in this special set of circumstances where, of course, people want maximum transparency, what is it that we can disclose without compromising the clinical trials and without compromising patient individual privacy?
Yes, so my understanding is that the trial has restarted now in the UK, Brazil, and South Africa. What about the US?
The US, we're waiting to hear from the FDA that is reviewing the data we submitted. Essentially, the data is reviewed by an independent safety committee that is then submitted to the regulator. And because the study is actually, in our case, the study is conducted by Oxford University. We are not the sponsor, they are the sponsor of the study. And they, of course, focused originally on providing all this information to the MHRA and to the regulators in the other countries where they are sponsor. We are the sponsor of the US study. So we then provided all this information to the FDA, and we're waiting to hear their decision.
Yes, and so you know, as we speak of transparency, you already mentioned the disclosure protocols. So of course, this is very unusual. You've disclosed protocols, as has Pfizer and Moderna, to other companies involved in vaccine development in phase 3 trials. And of course, yesterday in the press release, Johnson & Johnson also released the protocol. So why this move? It is unusual, certainly unusual to disclose the protocol before the study has concluded. Paul, perhaps you can comment on that.
Well, there's clearly, we are in very different and difficult circumstances where everyone is expecting the vaccine. It is having an enormous, the disease has an enormous impact on the world economy, and people, the political debate about it is like, are these guys doing the right work in order to get to the good information which tells that the vaccine works and is safe? And that's where I want to first come back to what Pascal was saying. In normal circumstances, we are very much already scrutinized by experts, the independent safety, the regulatory, the ethical committees in Europe, IRBs, all people with expertise and experience in 20, 30 years of vaccine development. So they have an oversight of what can happen in vaccines if you do experiments and new vaccines. They also know the very rare side effects which have appeared over the past. So they can get a good estimation of what is an adverse event, what is an unexpected adverse event, and make the judgment. On top of that, we have teams which are very specialized in certain neurology teams, we have inflammation teams, who all can do the interpretation of the data. But in this case, that's not enough. We have to be very open about what we do, the statistical designs, the endpoints, etc. And that's why we are committed to say, okay, let's make it available immediately. What Pascal was also saying, we can't disclose data in a placebo-controlled study on an individual level, because then you unblind the study and the study becomes void. So we have to have a careful way of how we work with the regulators, the review committees, the DSMBs, and with the general public on making sure they trust the process, they're well informed, but they don't hamper the process, because that doesn't help the public either. So when we jointly are committing to significant transparency up to the point that we can continue to do our work in a very professional and scientific way.
If I may add, Magdalena, is that we all want to be as transparent as possible, honestly, but the reality is, as Paul said, we have to make sure we protect patients' privacy, but also importantly, protect the study integrity. And at the end of the day, I think people people have to accept that they have to trust someone at some point, right? So you have independent safety committees, you have regulators, and you don't have one regulator. You have several regulators around the world. You have the FDA, the MHRA, the EMA, Japan. I mean, so many regulators will look at this data and these results with different eyes. So, I mean, at the end of the day, you got to trust that the experts whose job it is to monitor these trials and these developments are doing a good job. Medicine should not be practiced through the media. It should be practiced by experts, right?
Yes, you mentioned trust, and of course it's a sort of a limited commodity, especially today. Do you think, just following up what you said in answer to that question, do you think there's anything more that we could be doing to ensure or reassure the public that even though transparency is limited for reasons that you just very well explained, they ought to trust the experts, that they do have safety and indeed efficacy in mind?
Well, I think regulators, IRBs, DSMBs, they are not put together by just the normal people. They are always the collection of experienced, independent, not connected to the company, not connected to the process, as an independent review and overseeing what you do as a company. And we do that continuously in science. As you probably very well know, Maile, how you do this is you have your own process. You have an independent committee reviewing with experts what you do, giving advice. But in this case, they are the deciders. And DSMB can decide, the FDA they can decide. So the decision is not up to the company. We give it to external experts, which are very highly experienced in this matter, to say whether yes or no there is an issue. And I think the people should trust that process on that's happening, that is understood and very well understood, so that people can trust the whole situation as a whole on how we do developments in the world. And it's not different from cancer developments, inflammation developments, others. We always do this in pharma on how we develop medicines.
I mean, you know, even if people don't trust their own individual country regulator, there's also independent safety committees that are not subject to political pressure. I mean, those are independent people. They don't work for the government. They don't work for the company. They're totally independent experts. Point one. And point two is, you really would have to love conspiracy theories to believe that all regulators around the world will all agree to approve a vaccine that is not safe and effective. I mean, it's hard to believe that every country would do that. So you're going to have several set of eyes from different countries looking at this data.
Mm-hmm. Of course, clearly we so absolutely point taken. Clearly, there's some work to be done, because some recent polls, for example, in the US, showed that the percentage of the population who says they're prepared to take a vaccine has actually dropped since earlier this year to now, which is paradoxical, as we are perhaps getting closer to it. But speaking of timelines, let's talk a little bit about timelines. So some still hope that there may be an approval for emergency use this year, but realistically, when do you expect to have sufficient data to apply for regulatory approval? How long does that take? And assuming that the vaccine is approved, how long after that individuals such as me, let's say, could expect to be vaccinated?
I can start. We just started a phase 3, so we have to. It will take its time, which it will take to get to the endpoint on safety and efficacy. So we expect that somewhere around the year and first quarter, depending on the incidents and the run of the study, we'll be able to get to an endpoint which will show efficacy. But the endpoint in efficacy is not just the single thing. We also have put in our analysis plan that we have to have enough exposure to safety before we will stop the study. because there is no use in stopping for an endpoint for efficacy if you don't have extensive safety data, for example, in elderly and in certain populations. So the combination of efficacy and safety data will determine when we're there. But at the earliest towards the end of the year, but more reasonable, I think, in the first quarter, depending on the run of the study. If the vaccine is chosen for emergency use, because that's done by the government, that moment will be ready in January to deliver the first batches of vaccines. It will grow over the years. We have a commitment to 1 billion vaccines next year. We are pretty sure we can make that happen, but in the beginning, it will be limited, and it will be first going to the highest risk population, which are the healthcare workers and the elderly. Before you will get a vaccine might take some time in next year before the vaccine will reach you, Magdalena.
That's right. So there have been actually very recently at least a couple of recommendations as to prioritization of who in the population, the society should be first in line to receive a vaccine. But Pascal, do you want to add anything to this?
I think Paul described it very well. I think the one thing also that people tend to forget in terms of vaccination is you need results, but then you need also manufacturing. The day we have results doesn't mean the day after we can start vaccinating people. First of all, the regulators will have to look at it, look at the data and give approval, an emergency use approval in the US, for instance. But then you need to have enough quantities. And the key in this in epidemic is to be able to vaccinate as many people as possible around the world, starting with those at risk, of course, but covering the entire world. And it's a big population. So we will need several vaccines, and we will, of course, have to wait a little bit until we can scale up over time as an industry to get enough doses to vaccinate everybody who wants to be vaccinated.
Exactly. That's a very important point. A quick follow-up to this. So I believe the day before yesterday, the Washington Post wrote about the fact that the FDA is considering tightening its standards for emergency authorization of the COVID-19 vaccine. Do you consider this would be a significant setback?
I do not know yet the details. At least I might have missed it, but that's not yet communicated what that is. But there's talk that it will that they want to have the follow-up of 2 months after the cases. So that might add to the timeline or not. I have not yet looked into that, but it will not be a significant setback. It might add some time to the timeline, but that is to be evaluated as the regulations come out.
Okay. So I have a broad question to both of you as we coming to the end. We talked about the fact that we live in a very different special circumstances. You have released your protocols of your trials. You have pledged to develop emergency vaccine on a non-for-profit basis. We're talking about greater transparency, much more collaborative approach. Does this mean clinical trials in future will change?
I think we have learned on acceleration here. When it comes to real medical urgencies, I think the companies and the FDA are now very well trained. We did this before in cancer and in HIV and in emergency circumstances to go fast. We also have more access to data science now to see where to go for clinical trials. I think the collaborative spirit in the world between companies, but also between companies, regulators on how to interface in order to go fast is trained by this, is much better. So I see optimization of faster development of new medicines, new vaccines in the future, because we went through what we went through. It's a big learning on how to work 24/7 and being able to interface with scientists all over the world as well as with regulators. It teaches us a lot.
So on this very positive note, I'm afraid we're going to have to close because we've just run out of time. We could spend much more time talking about it. Thank you very much to Pascal and Paul. And there will be a second session this afternoon if you want to listen more about COVID-19 vaccine development. Thank you very much.